You're listening to Medscape's In Discussion series on colorectal cancer, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Benjamin Schlechter. Dr. Schlechter is an academic medical oncologist at Dana-Farber Cancer Institute and instructor in medicine at Harvard Medical School. Relevant disclosures can be found with the episode show notes on medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Benjamin Schlechter, and welcome to Medscape's In Discussion series in colorectal cancer. Today we're discussing HER2-Amplified Colorectal Cancer with Dr. John Strickler. You've led the conversation in colorectal cancer and the treatment of HER2-amplified disease, and your work has resulted in the recent approval of tucatinib with trastuzumab for HER2-amplified colorectal cancer in the metastatic setting. Before we get to this topic, I'd love to hear a bit more about your path to becoming a GI oncologist.
I did my medical oncology training between 2008 and 2011, and at that time, there was an explosive growth in this idea of precision cancer medicine, and I knew I wanted to be part of that growth of that field. It was not that long ago when there were no actionable targets in colorectal cancer, and the only thing we had when I was a trainee was KRAS exon 2, which was an inactionable target. We knew what not to give. So I was attracted to this idea of bringing precision cancer medicine to a difficult to treat disease where outcomes were poor and we were heavily reliant on conventional chemotherapy. And ultimately, I was hoping that with my career, we can move away from cytotoxic chemotherapy into more targeted approaches. Of course, we didn't even know about immune therapy at the time. I remember being a fellow and just seeing this presentation at ASCO about an immune therapy for melanoma, and I thought to myself, that will never come to us either. And now we can see how it's even applied to GI cancers. So for me, it was an opportunity to make an impact on a field that was still highly dependent on just traditional chemotherapy.
Yes, the immune therapy story is a really important one, and we do have a podcast with Dr. Faki from City of Hope on that very topic. So first, let's dive into HER2-amplified colorectal cancer. And I think the first thing we should do is define it. So how should we define HER2-positive colorectal cancer? And what testing should we be using? Is this an IHC test? Is this an NGS test? Where should we go? What should be introduced into practice at this point?
So in truth, there is no national standard for how to define HER2-positive disease. The traditional way that's tested in both gastric cancer and breast cancer is with immunohistochemistry in FISH. And there are specific criteria that have been developed to define that HER2 positivity for those two diseases, but we don't have the same adopted standard in colorectal cancer. There was an attempt in Europe to create a standard for colon cancer using IHC and FISH, but it never really caught on in the United States. Interestingly, when you use the gastric criteria or the breast criteria for colon cancer, the same applies, and we see pretty high concordance between those two different scoring methodologies in colorectal cancer. But the truth of the matter is, and the pragmatic fact is that we're generally testing for HER2 out of our next generation sequencing tests. And that's how we're usually finding it these days. And it appears that there's fairly high agreement between the classic IHC and FISH assays that we're accustomed to using gastric and breast criteria and the next-gen sequencing profile. So it is ambiguous how it's defined, but the good news here is it really doesn't matter at the end of the day. If you use a standardized CLIA-certified assay and it says it's HER2 amplified or overexpressed, odds are pretty good that patient will benefit from anti-HER2 therapies.
And do we know if there's a distinction between tissue-based assays versus ctDNA, for example?
There is an important distinction. So ctDNA, the commercial tests we use are also next-generation sequencing tests, but they are obviously based on fragments of DNA detected in blood. And it is somewhat trickier to detect an amplification in blood than in tissue. And the interesting thing is you need more what we call shedding or a higher tumor fraction to detect amplification in blood than say you would with a typical KRAS mutation. And that can make it tricky. So you can trust a positive result. If it says HER2 amplified by a ctDNA test, it's probably HER2 amplified. But if it's not amplified in blood, I would still revert back to tissue testing to confirm that it's not amplified in tissue. So you can trust a positive, but you really can't trust a negative.
Excellent. That's really helpful. That seems to be the story with ctDNA. Tissue testing still has legs, it looks like. And do we know of any differences left-sided versus right-sided? Clearly, this is a big story in BRAF and KRAS. Are there differences in HER2?
Yeah, so it's a really interesting question. There are some features to HER2-positive disease and colon cancer. Number one, it has a much more homogeneous staining pattern by the traditional IHC criteria. So it kind of stains a little bit like a breast cancer, which will be more homogeneous. Gastric will be more heterogeneous. It tends to be on the left side of the colon, not the right side. So about maybe around 80% of these HER2-positive colon cancers will be left-sided. And those are some of the unique features of it. It also will tend to be on the IHC 3+ side of things. It's not very common to see a 2+ FISH positive, tends to be at that higher level of expression. So there are some unique features to that, and I think that explains why these anti-HER2 therapies are so active for HER2-positive colon cancer.
Let's talk about the MOUNTAINEER study. This was, I think, a big splash, and people were really excited to see the results. So can you tell us the background of that trial and, frankly, what you did and what you found?
Well, I'll go all the way back to when I joined faculty. When you join faculty at an academic medical center, you kind of feel like you're clinging to the cliffside. Am I going to make it in academia? And my mentor at the time, Herb Hurwitz, gave me a little trial, first in human phase 1 study of an anti-c-Met antibody called ABT-700. And we struggled to find these patients who had MET amplification. And one of the things we found out was that this tended to be easier to detect in blood as a resistance alteration than tissue. So we made a partnership with a little company at the time that turned out to be Guardant Health. And we started to look for MET amplification in blood. And I was looking at these patients with RAS wild-type metastatic colon cancer and finding that a number of these patients actually had HER2 amplification in blood, not MET amplification. And because at the time we were not testing for HER2, I would go back and look at the tissue by breast criteria, and sure enough, they were HER2 all along. So there were these grumblings that maybe there were these HER2 therapies would be active. And at the time, I was seeing some very intriguing data about tucatinib and trastuzumab, actually in combination with capecitabine. And I wrote up a little one-page proposal for a tiny company in Seattle called Cascadian. And I said, hey, your drug is new in the clinic, very active, very selective, very well tolerated. Let's see if it's active for HER2-positive colon cancer. And they got back to me in about an hour and said, let's talk about this. Let's see if we can put this together as a pilot. And it just took off from there and was a ton of fun to run. And, special thank you to colleagues. This was an investigator-initiated national study. I called up all my friends, including at your institution, Ben, and we all just pitched in as investigators. And I also want to give a special thank you to the patient advocacy groups. There's a group on Facebook called Colon Town, and they took this trial under their wing as well and really got the word out there for this pretty rare alteration to say, hey, if you know anybody in the United States who has this, here's this amazing trial. It was an example of investigators working together as a team, patients and patient advocacy groups working together as a team, and industry really just giving us the tools we needed to make it happen. It was a lot of fun to get that off the ground.
That's such a great story. And it's funny you call it rare. I was thinking about the incidence of HER2-amplified colorectal cancer compared to, say, MMRD advanced colorectal cancer. And how do they compare? Because when there's a big splash in mismatch repair deficient disease, this is front page news.
Absolutely. They're actually pretty similar in terms of their prevalence. And so we see HER2 amplified or HER2 positive disease in about 3% of patients. And it kind of depends what data set you look at when you look at mismatch repair deficient colon cancer, but that tends to be in the 3 to 5% range as well. They're actually pretty comparable in terms of how often you'll see it. The good news is we test for both out of our classic next generation sequencing panels. I think this really hammers home that the moment you see a patient with newly diagnosed metastatic colon cancer, just order that next-gen panel. It will test for MSI, it will test for HER2 amplification, and then, of course, all the more common variants like KRAS, NRAS mutations. Even BRAF mutations are almost similar in frequency to HER2 amplification.
So now back to MOUNTAINEER. So what exactly did you find with this tucatinib combination?
We initially presented the first results of what we call Cohort A, that's tucatinib and trastuzumab, at ESMO in 2019 and posted a very impressive response rate, 46%. And based on that result, we knew that this had the potential to become an FDA-approved standard of care one day. So we transferred the IND over to the company. Cascadian was acquired by Seagen, and they took it forward and turned it into a global study. And in those combined cohorts A plus B, the confirmed objective response rate was 38%, progression-free survival over 8 months, and overall survival over 2 years. What was particularly impressive is the duration of responses. When patients responded, they responded for a long time. And in fact, the very first patient that we enrolled is still, thankfully, doing great today. And this is somebody who enrolled many years ago. And we have durable, complete responses even today. So we see that in these very high durability of response numbers. So it's an exciting breakthrough for our patients. And I really appreciate the fact that it's a chemo-free regimen. That was the goal at the very beginning.
How's it tolerated compared to chemo? How are people doing on tucatinib trastuzumab?
Thankfully, most patients do fantastic with it. So the strength of this regimen is that it's highly selective for HER2. So you do see some diarrhea in about half of patients. Most patients describe it more as a nuisance, not really intolerable. It'll pop up every couple days, and it's usually well controlled with Imodium. Occasionally, you'll see a rash with it. Very rare cases of liver enzyme elevations that will lead to discontinuation. That's probably the one thing that will lead to a treatment discontinuation. We only saw that in single-digit rates, very low number of patients on MOUNTAINEER. But that's the main toxicity to look out for. And I think for a patient who's had a lot of exposure to chemotherapy, this will come as a very welcome respite.
Yeah, I have to say I have a patient who's still on, as she's 2 years in. And in the beginning, she kept saying, "Are you sure I'm not getting the placebo?" And I said, "There is no placebo." So I know exactly what you mean. I have seen some diarrhea as well, but overall, I agree. Very well tolerated.
Yeah. In fact, the one side effect that we were commenting on that we found to be one of the more common ones was weight gain, because people felt so great. They were eating and living life, and all that weight that they lost with their cancer diagnosis came right back.
Yeah, we need to add that to the quality assessments, right? Like weight gain is a sign of quality of life.
Absolutely.
The other sort of big innovation, HER2-directed therapy or antibody-drug conjugates, and in particular, trastuzumab deruxtecan in colorectal cancer, as well as gastric cancer and breast cancer, even a standing ovation for HER2-low last year in breast cancer at ASCO. So where does that fit into the paradigm for colorectal cancer? How do we give it? How do we dose it? How does it stand?
You're referring to trastuzumab deruxtecan, which is also a breakthrough for our patients with HER2-positive colon cancer and something that I've used in the clinic as well. And just as a reminder, DESTINY-CRC01 was presented, was it 2 years ago now? And there was a recent paper that just updated results from DESTINY-CRC01. So impressive response rate out of that initial cohort, 45%, but there were some interesting signals from that cohort. First, almost all the responses occurred in patients with IHC3+, or the highest level of expression. I believe there was only one response in patients with IHC2+. So it seemed, unlike the story we heard from last year at ASCO and HER2 intermediate and low breast cancer, it seemed like all the benefit was concentrated in patients with very high expression. Additionally, there was a very concerning signal of grade 5 or fatal interstitial lung disease or pneumonitis. And now that data has been updated. 3 patients in that initial series have died, and that unfortunately reflects an unacceptably high death rate from that 6.4 mg per kg dose. So I think it was really those 2 issues that inspired the next trial, which was DESTINY-CRC02, which we just saw presented at ASCO a little more than a week ago, to randomize patients to either the higher dose, 6.4 mg per kg, or the lower dose at 5.4, to really answer that question: what's the safest dose? What's the most active dosing? The results came as a surprise. Number one, the activity was not reduced with that lower dose of 5.4 mg per kg. We actually saw a slightly higher dose, a higher response rate at that lower dose, 38% response rate. Secondly, once again, all of the responses, save one, occurred in patients with IHC3+ disease. Another really important finding and unmet need is patients with RAS mutations appeared to experience significant benefit from this trastuzumab deruxtecan drug. So now we have an option for those patients who have both HER2-positive disease along with a RAS mutation. So really important findings from that. And then finally, once again, we saw this signal from DESTINY-CRC01, but it was confirmed in this latest DESTINY-CRC02. Response rate appears to be identical whether or not you've had prior anti-HER2 therapies. This lays out a story for us of how to manage these patients in our clinic. And the lesson is, number one, test for HER2 right out of the gate. treat those patients with upfront chemotherapy and bevacizumab. Then, when they are in the refractory setting, if they have RAS wild-type HER2-positive disease, start with tucatinib trastuzumab. And when they progress, then you have trastuzumab deruxtecan as a backup option. If they have HER2-positive disease but a RAS mutation, go straight to trastuzumab deruxtecan. So that will be kind of our emerging paradigm for how to manage these patients.
Excellent. That is just a super helpful summary and like a great story for how we landed here. And when you see this pulmonary toxicity from trastuzumab deruxtecan, how are you treating it? I know we are sort of lacking in prospective data. I'm just curious to hear your particular approach here.
When this drug came into the clinic, we turned to our breast oncology colleagues and said, how are you guys managing this? Number one, they were using the lower dose to start with, 5.4 mg per kg. And at the time, I was uncomfortable with the high rates that they saw in DESTINY-CRC01. So I always adopted that lower breast dose, 5.4 mg per kg. They also have a protocol here that we developed at our institution that we do a baseline pulmonary function test, and then we check PFTs every 6 weeks for the first few months. And we're looking for drop in this DLCO, right? We're looking for signs of pneumonitis or ILD. And we have consults built up with our pulmonary service will support us if we see that drop in diffusion capacity. So that's how we manage it at Duke. Knock on wood, I've not had problems with trastuzumab deruxtecan yet, but unfortunately it is a known toxicity of this drug and it's something we have to be very careful about.
What about the cardiac signal? I remember from fellowship with trastuzumab and all the cardiac monitoring, are we seeing cardiac issues in the GI population? Are they living long enough for that?
Historically for us, Ben, the main cardiac issues we have are cardiovascular disease, coronary artery disease, which limits our ability to give bevacizumab. Our patient population tends to be a little bit higher risk for coronary cardiovascular disease. And so sometimes I've stumbled into cardiovascular disease just by virtue of the standard every 3-month echoes. We did see a couple of asymptomatic left ventricular ejection fraction drops on the MOUNTAINEER study. Fortunately, they were asymptomatic and reversible, but it does not appear that we see higher rates of these left ventricular ejection fraction drops in our population of HER2-positive colon cancer than, say, in the breast population. So I think, fortunately, the cardiac signal has not been an issue, at least in my experience so far. I don't know if in your experience, if you've had any difficulties with it.
No, I really haven't. I've had the same experience as you, being really diligent about pulmonary toxicity, but I've not seen any cardiac toxicity in my personal patient population. It's a really tour de force, a very impressive effort in HER2 and a fantastic summary. So you seem to pick your targets well. So what are the other targets we should be looking at? What's coming forward?
Fortunately, there are more targets. And I think the first nut that we are in the process of cracking right now is KRAS G12C, which interestingly is almost identical rates in colon cancer as, say, HER2. So about 3% of colon cancer have this KRAS G12C alteration. It is highly targetable with the dual KRAS G12C anti-EGFR combinations. We've seen publications in New England Journal and presented at multiple conferences. So I suspect that combination will enter the clinic sooner than we might expect, potentially in the next year. We'll see when that data comes out and we see an FDA approval. So that's exciting. I'm also, as a targeted therapy proponent, very interested in KRAS G12D. I do think we're on the cusp of some important therapies coming into the clinic to target KRAS G12D. Really fascinating chemistry behind this. It's not an easy target. There's a reason why it's taken so long to get to this one. And another really interesting class that I'm curious about are the pan-KRAS inhibitors. KRAS G12D is one of the more common alterations we see in the clinic, but we see G12R, we G12V, we see other KRAS variants. So would love to get my hands on those as well one day and bring those to the clinic. And almost certainly they'll be combined with an anti-EGFR therapy to deepen the responses and lengthen those responses.
Excellent. Yeah, I'm looking forward to that too. You know, the change to a neutral amino acid to a polar one we can target, but these other changes are going to be hard. Looking forward to hearing you come join us and talk about it.
Yeah, I can't wait.
Today, we've been discussing HER2-amplified colorectal cancer with Dr. John Strickler, and we discussed that HER2 testing should be considered in all patients with advanced colorectal cancer, particularly next-generation sequencing options, which should be applied for all patients with advanced disease should identify this. Tissue-based assays are critical because circulating tumor DNA may miss it. IHC is also important, but the 3+ disease seems to be the population that derives the most benefit from this. We reviewed the data from the MOUNTAINEER study, which Dr. Strickler led, and this showed that the combination of tucatinib with trastuzumab has a high response rate and durable responses, and RAS wild-type HER2-amplified cancers with a manageable safety profile, really just some minor GI toxicity and some transaminase elevations, which patients can tolerate with a non-chemotherapy approach. We also discussed the antibody-drug conjugate of trastuzumab deruxtecan from the DESTINY-CRC studies, and we showed that 5.4 milligram per kilogram is both safer and as effective as the higher dose regimen to mitigate the risk of dangerous pulmonary toxicity, as well as concerns over cytopenias we see with other chemotherapies. And then finally, we talked about some promising targets, in particular KRAS, which is, I think, in many ways, the holy grail of colorectal cancer as it stands now. So thank you for joining us. This is Dr. Ben Schlechter from In Discussion.
Thanks for listening to Medscape's In Discussion Colorectal Cancer podcast series with our host, Dr. Benjamin Schlechter. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on colorectal cancer.